Hi, everyone, and thanks for joining us for episode 702 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am looking forward to re-sharing our interview with Dr. Adam Abate, originally published on January 30th of 2017. Listeners, Adam is a physicist by training, and in his lab, he and his team are developing technologies focusing on microfluidics and high-throughput biological technologies to allow us to comprehensively characterize cells in a system. He uses these approaches to solve problems and answer questions in the areas of evolution, genetic sequencing, and cell sorting. And in our interview, Adam is going to share some great stories from his life and research. So listeners, get ready for this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, our wonderful guest is Dr. Adam Abate. So, Adam, welcome to the show. How are you?
Doing well today.
Excellent, excellent. Well, we are delighted to have you with us, and I'm excited to introduce you to our listeners. So, listeners, Adam is an associate professor in the Department of Bioengineering and Therapeutic Sciences at the University of California, San Francisco. He is also a co-founder of the startup company GnuBIO. And Adam received his bachelor's in physics from Harvard College, his master's in physics from the University of California, Los Angeles, and his PhD in physics from the University of Pennsylvania. Afterwards, Adam conducted postdoctoral research in physics and engineering at Harvard, and during this time, his research became the foundation for the sequencing company GnuBIO. And Adam is currently a member of the California Institute for Quantitative Biosciences, or QB3, and this is a program that helps launch startup companies on the UC campuses. Now, Adam has received a number of awards and honors during his career, including the NSF CAREER Award, the NIH New Innovator Award, and the Presidential Early Career Award. And listeners, Adam is here with us today to talk about his research and to tell us about his experiences in life and science. So, Adam, today we are excited to get to know you both as a person and also as a scientist. So can you start by telling us what you like to do when you're not doing science?
Well, actually, it sort of changes every few months. At the moment, my new weekend hobby is basically home improvement. My wife and I, we just bought our first home about a year ago, and it's definitely a fixer. So neither of us really grew up doing home improvement kind of stuff, but it turns out actually if you're pretty good at the bench, you turn out to be, I think, pretty good at fixing wiring and so on. So recently, our weekends have just been installing lighting and painting and that sort of thing. And it's been interesting and fun because it's a new challenge. And unlike science, where so much of what you do ends up basically as a failure, right? You fail so much to have a few amazing successes. When you're working on your house, you see steady progress for every hour that you put in, and it's very gratifying and just another challenge. So that's pretty much all I've been doing when I'm not working is working on the house.
Well, I definitely know that feeling. We've almost renovated our entire house ourselves, and it is absolutely so gratifying to see the progress. You put that hour of work in and you can immediately see this room looks better, which is not always the case in science.
Exactly. I think I have a talent for it because I'm so used to having absolutely nothing to show for all of my effort. Having such instant gratification is really nice.
Absolutely. So what is the current room you're working on now?
So we just actually installed lighting in our kitchen because the house had one of those wonderful dropped ceilings that they built in the 60s or 70s. So we took that out and, of course, had no lighting in our kitchen for a few months. So I had to figure out how to cut holes in the ceiling and install lights. And, it turned out to be actually really easy. The hard part is actually understanding how electricians talk about electronics versus scientists. And once you make that connection, it ends up being pretty straightforward how to do things.
Awesome. Well, great to hear a little bit more about your life outside of science. And I think this kind of fixer-upper weekend hobby is a great way to balance that maybe extreme lack of instant gratification in the life of a scientist. So,
yeah.
So now I'd love to hear more about your science next. So Adam, can you tell us how you describe your work to someone who's not at all familiar with your field?
I'd say that overall, sort of the big motivation of what we do in my lab is to try to basically to make biology a new kind of computer science. In other words, like a computer, biology has programs, but those programs are written in the form of molecules, DNA and protein. and other kinds of small molecules that are interacting. And the actual system, the actual computer, is a physics engine, which is the actual real world. And that kind of idea is really at the basis of different fields, systems biology, synthetic biology. And it's really this idea that you can think of biology as a system that translates information, genetic information, into substance, to phenotypes, to cells, living organisms. It turns out, of course, that all organisms are incredibly complicated. And this is why biology is one of the most difficult disciplines, is because the systems are so complicated that they often seem totally unpredictable. And as a result, much of the science is reductionist in nature, where you try to simplify and extract meaning that hopefully is somewhat general. But ultimately, my belief is that if we want to really be able to understand biology and manipulate biology, we have to think of it sort of like we would think of a computer. You have to be able to characterize its state in detail and ideally understand that and be able to manipulate it to create the outcome that you want. And there's many examples you could think of. One is curing a disease that somebody has. Every disease is basically some problem with the state of the system. And our goal as researchers or doctors is to figure out how to correct that problem. But that's a very difficult challenge in general because these systems are so complicated that it's not at all obvious what changes you have to make to get the system back to where you want it. So that's changing in my view. And I think there's going to be a massive transformation in the next 20 years in biology because we are now coming to a point where we're starting to have the hardware, the actual experimental tools where we can write down what the state of a system is completely. I mean, we're still not there, but I think we will be there in the next 10 years or so. And once we have that ability to write down what a biological system is, the hope for me is that we'll be able to actually understand how to manipulate it and control it. And so where my lab focuses specifically is on building the tools that allow us to characterize those cell states very comprehensively and with complete detail, or at least as complete detail as you can get. So specifically, we work in a lot of what's called single cell analysis, where you think of very complicated biological systems like a human being as basically a collection of cells that are all running these programs, these genetic programs to create this organism. And so there's a lot of interest in my field in actually sort of breaking up that organism into its parts and characterizing those parts in detail. It's impossible for a human being to ever do that characterization manually. Obviously, we're composed of trillions and trillions of cells. So we need automated technologies to do that. And my lab focuses on building those kinds of automated technologies, much of it based on microfluidic technologies and software that goes along with analyzing that data.
Well, that is phenomenal. It sounds like we are at a really exciting point within your field where all of these pieces are now starting to finally fall into place.
Yeah. It's really driven by the advances in computer technology, which has then enabled things like high-throughput sequencing and mass spec, which are tools that biologists use to extract huge amounts of information about a system. And so we now have a way to create huge databases to characterize these systems. And those are still just a tiny fraction of even the most simplest biological system. So I think it's going to expand. And then we're going to have another problem, which is how do you actually take this information and understand it to figure out how you can change the system and make it have a property that you want.
Absolutely. Well, Adam, thank you so much for telling us a little bit about your work, and we'll chat more about your projects as we go through our interview. But I want to talk a little bit first about motivation. I think this is a really important force in the life of a scientist. You have to be motivated to go in every day, despite a lot of these failures that you're facing that we mentioned. So can you tell us what motivates you, or do you have a favorite quote or saying that you use to keep yourself going?
Sort of. I saw a documentary about Steve Jobs some years ago, and what stuck in my mind, I don't remember exactly how he put it, but he said the realization he had at some point when he was pretty young was that all the incredible technologies that you see around you, they weren't built by superhumans or super intelligent beings. They were built by regular people, just like you, no smarter. And I think that is one motivational quote. The reality is that each of us has the ability to understand the natural world, and that's a gift, if you think about it, that's been given to the human species that we all have. And I think when so much of science is just unrelenting failure, remembering that you do it for those rare instances when you get to learn something new or discover something or invent something useful because you have that ability, that keeps me going. And another thing is just to remember to be grateful for the opportunity. It really is an amazing opportunity to be a scientist. Our work is funded by the generosity of the taxpayers. And I think just recognizing that and being grateful for the opportunity to be able to go after your own ideas and follow your own intuition, even if you fail, it makes it a little less painful because you just recognize that it's just such an awesome opportunity to be able to even try. So many people don't have that opportunity. So those kinds of the things that keep me going when we have the 9th failed experiment in a row.
Of course. Well, I think even when you fail, you often do still learn something. So it's not a complete waste. So.
Oh, absolutely. Absolutely.
Well, Adam, wonderful to hear more about what motivates you. And I'm curious to hear about some of these people who you've encountered along your journey who might have inspired you to pursue science or served as mentors or role models. So does anyone in particular come to mind for you?
Yeah, I've had many role models from my 9th grade biology teacher, Miss Spina, to my role models in grad schools. I had a really excellent PhD mentor, Doug Durian. He's a physicist at Penn. And I would say under his tutelage is where I learned to think like an adult, to think carefully and methodologically. He's a very careful and extremely intelligent physicist, and working with him really taught me how to think, basically. And then after that, I went to work with a guy named Dave Waits at Harvard, who's just the most awesome person you can meet. Everybody loves him. All of his students and postdocs love him. I really consider him as much more than just a mentor. He's a friend and a role model, someone who I really do try to model myself after. And one of the things in particular that I've always appreciated with Dave is that I sort of consider myself an intuitive thinker, and he is also a very intuitive thinker and has an incredible ability to make things that seem at first extremely complicated and just incomprehensible. He makes them seem really simple, and he explains them to you so that they're simple. And the amazing thing is that most of the time you remember them as being simple. So sometimes you meet these people that are just really great at just explaining things and being charismatic. But then about an hour after you talk to them, you can't quite remember what the point was, or that ability doesn't transition to you. And with Dave, he's such an intuitive teacher and mentor that many of those abilities, if you're just observant, he's able to pass them on. And he's been wildly successful as an independent scientist, but also in terms of the number of faculty that have spawned out of his lab is just huge. So he's been an important person in my life scientifically. And then the third is someone I've only ever really observed from afar, is another guy, Steve Quake at Stanford. He's sort of like me as my trace through science, where he started out in physics and then transferred to more biological sciences. And he's just been such an incredible success, making that transition, doing really groundbreaking research, and then also taking that into extreme success in the commercial sector. So he's created many companies and been very successful there. Actually, I know Steve much more now because we're both in the Bay Area, but even before that, I was just sort of in awe of his career. And he was an inspiration and in some sense a motivation to take this track myself, because it is definitely pretty intimidating to be a physicist and to ultimately end up becoming a professor at a medical research university. And seeing someone like Steve who was able to do that so successfully had a pretty big impact on my career. So I'd say those are my 3 biggest mentors that have really affected my career to date, and who I still really try to talk to as often as possible.
Excellent. Well, Adam, it sounds like you've had some really key people influence you at these maybe important points in your development as a scientist. And I think that can make all the difference in terms of the path that you take afterwards. But can you remember all the way back to the very beginning and tell us what first got you interested in science?
So I don't come from a very scientific family. My parents were both just regular people, regular jobs. My mom is actually an immigrant from Honduras, and science wasn't a big part of our lives. But it turned out when I was a kid, I met another kid named Glenn, who's a really good friend of mine today. I think we're naturally scientists, just the way we think. And we became really good friends, and he was really into math and physics, and that sort of got me more into math and physics. And then we took all the advanced classes and then the AP classes and literally ran out of advanced placement classes we could take in college. And then we ended up both going to Harvard for undergrad together. So that was a pretty transformational time, I think, in my development. And then once I went to grad school, working with Doug and then Dave, of course, those were pretty important too in my career. So that's sort of my trace through time. I think for me, the big question I always had was, do I stick with science or do I go and do something else in the private sector like many of my friends have done? For me at the time, that was a very easy decision. I knew I always wanted to be a professor, but it does come with cost. You don't make nearly as much money, of course, but the trade-off is that you get to do whatever you want, right? You get to, well, not whatever you want.
Right.
But you get to do largely what you want and follow your own ideas, which is really an amazing privilege.
And now you straddle the entrepreneurial and academic world. So can you tell us a little bit about what that balance is like?
It's hard. I think as a technologist, someone who builds technologies, really the reason we're doing it, it's not just to learn something, right? It's not just basic science, as people would say. It is a large part of that, but it's also to make an impact. And the way that we make impacts as technologists is that we get our technologies out there into the real world where they can actually improve people's lives. But the problem that you have is that in many ways, the overriding philosophy of academic research is almost the opposite of the philosophy of commercialization. Mm-hmm. One is really about being open and following your intuition and doing it just for the sake of doing it, for answering questions. And the other, it has to be about making money. And those two things are difficult to straddle because they're just so opposite in what really motivates why you're in those two fields. But I do think that it's really a responsibility of technology developers like myself, because writing papers is great. But if you really do develop something or discover something really valuable that has commercial value, there's really no one else better than you, the person who developed it, to actually commercialize it. And it's very challenging, and it just takes relying on a lot of people who have different skill sets from you. I've been very fortunate where at UCSF, we have some excellent programs that have connected me with people that have a more business background. And I really try to go into these discussions with these people as humbly as possible, recognizing that I know really nothing about business and commercialization and trying to learn from them. But that's difficult because I think deep down I'm a scientist and I do things because I think they're interesting. And then figuring out how to connect that up with a way to make money is not something they teach you in grad school. And there really is no answer to that. It's something that you just sort of have to figure out as a case-by-case basis. And that can be tough. It's a lot of work, and it places a lot of additional work on my plate for thinking about how to make something commercially viable after you make a discovery. So from the point of just filing a patent, which can end up being a lot of work, to then doing the many meetings with potential employees or administrators or funders of the company. It's just a lot of work that you're really not trained for as a scientist. But at the same time, being the person who developed the method or invented the technology, it really is ultimately your responsibility to make it happen. It's a challenge. It's not something that I'll ever give up. I think I have to continue doing it, and it's been a lot of fun. But it's definitely a tightrope to walk because of that fundamental discrepancy between the motivations of the two fields.
Certainly. So do you have any advice maybe for young scientists who are out there who are interested in developing their own startup company, or maybe any tips from your own experiences?
It's sort of cliche from the business perspective, but recognize that as a scientist, your technology is just literally a small fraction of what's required to become ultimately a successful business. As humans, I think we always tend to overemphasize the importance of what we do, what we know, right? And we underestimate the importance of all the things we're ignorant of. And when you go into commercialization, I think it's really critically important to recognize that the technology, which you think is everything, is really not everything. It really is a small fraction. It's the minority fraction of what will ultimately lead to the success of that company. I mean, in most cases, companies are not going to even continue doing what the company was founded to do originally by the founder. They'll end up, what they say, pivoting into a new idea. So I think just being humble and recognizing that, and a strategy to addressing that is pair up with people who can fill in the weaknesses that you have. So find people that have a more business background and make them equal partners with you, because they are at least as important as you are to the success of your venture. If you're really serious about doing it, I think you have to, as soon as possible, you go out there and find partners with a business background who you can get them excited about it, and then we'll go with you hand in hand to actually take it forward.
Certainly. You've got to build that dream team with all the different skills. So,
yeah.
Well, Adam, thank you so much for sharing more about your experiences, more about your career path. And I'm curious to hear more about one of these projects that you have going on now that you are so excited to be working on. So can you tell us about, we'll call it your favorite?
Well, it's hard to choose favorites. We have so many cool projects going on in the lab, but one in particular, I think, goes back to this broad vision of my lab, something that we've been working for 4 years now. Feels like a long time. But this idea of being able to take a very complicated biological system and just write down the sort of software that encodes it. So in other words, the genomes of the cells that comprise a system. So if you just take a piece of crud off of the bottom of your shoe and you look at it under the microscope, you're going to find literally billions of different organisms, single-celled microbes. Every one of them is a unique cell. and can have a completely different genome from the vast majority of others in there. And so if you want to understand this ecosystem that occurs at this tiny scale, you would like to be able to just, as a first thing, write down what the different genomes of all of those cells are. Literally sequencing, what we call sequencing the genomes or storing the genomes of all those cells. There's many other things you'd want to do beyond that, but that's sort of the first thing I would want to do. And we've recently got that working. I had a really superstar graduate student, Freeman, who worked on this project for 4 years during his PhD. And he took it from this crazy idea that I had to actually something that worked in practice, where now we can take cells from a sample, a microbial sample, and we can sequence every cell in that sample individually. Ultimately, the process of doing that is a hardware problem. How do you take these millions of cells, isolate them, and then process them through this machine, a sequencing machine, so that you can recover the genomes as single intact genomes? And so Freeman developed this technology that actually does that. We have it working. We've done per run, we do about 50,000 single cells. So it's still not the billions that are there, but it's a lot more than anyone had done previously, where people were just doing literally tens of cells at a time by very low throughput methods. So I'm very excited about that. We have a paper already published and another one that is under review currently. And I'm really excited about taking that forward, where we now want to add on additional sort of dimensions to the analysis. So the genome is one piece of information that cells contain, but they also have something called RNA, which is another form of information in cells. And they also have proteins and they have molecules. They're this incredibly complicated interacting system of all of these things: DNA, RNA, protein, and molecules. And one of the things we're now working towards is adding on additional layers of characterization. So in addition to genomes, we also want to start characterizing the RNAs in the cells and their proteins, and eventually their metabolomes, the small molecules that they use to live. So that's a really exciting project. It's probably going to span the next 10 years of my career, but we've made our first big success in characterizing that most basic form of information, the genetic code of the cells.
Well, that is phenomenal. Do you think we'll ever get to a point where we'll be able to get into the billions of cells at a time being analyzed in parallel?
So we've done about 50,000 cells, but I do think we'll get there. In fact, the system we have now can probably scale to tens of millions if we pushed it. The problem is that we need to use the existing sequencing machines. So like everything, you ultimately are going to rely on some other tool to complete your experiment. At the end of the day, we do our microfluidic processing and we get a huge amount of DNA that we then have to put on a sequencer. And right now, the sequencer is the bottleneck. So we can produce the DNA and get it ready in a way where we can get all this information out, but it has to go into the sequencing machine. And right now, doing beyond tens of thousands of cells is just not feasible. But sequencers are rapidly increasing in their capacity, sort of at the similar rate to the rate at which computers are advancing, actually for many of the same reasons, because they're based on sort of miniaturization of electronics and so on. So they're getting a higher throughput, just like computers are. And so I would expect that in the next 10 years, we'll probably be able to do several orders of magnitude more sequencing than we can do today. And then with the technologies that we've developed, we will be able to get to billions of cells. But you have to recognize that's still just one piece of crud on the bottom of your shoe, right?
Right.
You go to the ocean, now you have the whole ocean of these microbes present, and it changes by the ecosystem. So the challenge is really, truly monumental, the amount of analysis that's required to characterize any of these systems. But I do think that is ultimately what will be required to really understand a biological system, because unlike most physical systems that physicists study, the properties of a biological system really emerge as a result of the complexity. They can't be understood
in isolation,
Yeah, by ignoring the complexity. And that's why biology is so hard. And so we have no choice. We have to build these technologies that allow us to characterize this complexity.
Certainly. Well, Adam, it's exciting to hear more about how your technology allows for some of these amazing advances in understanding these biological systems. And I think, like you said, it's probably going to take the next 10 years of your career. I think we have so far to go, but we are making amazing strides towards that point of being able to really have a grasp of these large, very complex biological systems. And that's phenomenal. But I think not everything about a scientist's job is amazing and phenomenal. We all have down days. There are challenges, struggles, and failures. And I love talking about these on our program because I think it's so important for people to really have a view of holistically the life of a scientist, the ups and the downs. So can you share with us one of your own stories of struggle or challenge or failure and how you work through it?
At this point, I'm sort of immune to failure because it's such a common part of the job. But I wouldn't say it's failure, but definitely struggle. just the whole career arc of ultimately becoming a scientist who's funded by government money, it's a very challenging and continuous struggle. You go into science because you have a love of discovery, understanding how the world works, right? That drives you to get a PhD, for example. And then you start thinking about, well, I want to eventually be able to have my own lab, of course, right? So now I need to start applying for jobs. And it turns out that the responsibilities that you have as a faculty member are pretty different from the ones you have as a grad student. What makes you good at a grad student doesn't necessarily translate over well to being a faculty member. Mm-hmm. I think good grad students tend to be very single-minded and motivated to get tasks done. And that helps for faculty too. But the fact is that as a scientist where you're managing people, so many balls that you constantly have in the air that you have to be able to switch between tasks rapidly and efficiently. And that's just challenging because I think the point is that the training that you get as a young scientist isn't the only training. It's not even close to the only training you need as a more senior scientist. And it's a continuous process of just getting better at handling all the tasks that are constantly being thrust upon you as a faculty member. And then, of course, fundraising is no fun. If there's anything more failure prone than science, it's grant writing.
Right. Which is unfortunately an integral part of science.
It is an absolutely integral. You can't do anything without money. You can't do anything without collaborators. There's not enough money in science, frankly, and there's just too many great ideas that scientists have. And it's extremely hard for the review panels to figure out who's going to get this precious money to do their science that they want. And it puts back on the scientist, it puts a lot of pressure, obviously, to write your grants, because unless you write an absolutely stellar grant, you have no chance. And even if you do, you only have a small chance. So you have to get used to a lot of rejection, and that's just a continuous challenge. But again, I think being able to follow my own ideas makes up for all of it.
Absolutely.
Sort of a scientific failure. I wouldn't call it a failure, but it's sort of another struggle we've had in my lab, and I think this is pretty common in sciences, where you need to understand something, okay? And you read in the literature and you see what people know about this, and you come away from that thinking, oh, well, people already understand this phenomenon, or they understand how this thing works. But you realize that actually it's wrong. The understanding is wrong. Mm-hmm. But it often is the case that the phenomenon, especially when you're developing technology, is not something people are going to get excited about. Like it's a fairly mundane thing that's absolutely critical to getting the technology to work. Like maybe a chemical that everyone thinks is necessary turns out to be not exactly the right chemical. So you can't progress without it. You have to figure out what the right chemical is. But if you figure that out, no one's going to really recognize that it's a huge breakthrough because people already assumed that it could be done.
Gotcha.
This is pretty common in science where there's problems that people think are finished, but they're not. And you can't make progress until it's finished because the reality is the world doesn't care what you think. It just is what it is, and you have to do it right and understand it properly. And so we've had many things like that where we read in the literature and we thought something was finished and it wasn't. And so we ended up having to do literally years of work to figure this out properly so that we could just do what people said was possible earlier. And one example was that actually in our single-cell sequencing technology that I described earlier, one of the things you have to do is you have to do a biological reaction on a single cell in order to prepare the genome so that it can go into the sequencing machine. We use PCR, polymerase chain reaction, which is something that's been around for a long time. But the methods that people described actually don't work very well. But no one had recognized that. They said it worked, and there was papers showing that it worked, but when we did it, it barely worked. We couldn't get any useful information out of our sequencers. So we had to spend about 2 years optimizing doing PCR on single cells in order to produce that material. And we wrote those papers, and they're reasonably impactful. Some people recognize it, but at the end of the day, I think many reviewers will look at that, and many scientists will look at that and say, well, people had already talked about doing this. This isn't really novel.
You're not going to get the Nobel Prize for it, yeah.
Yeah, you're not going to get the Nobel Prize for it. But it was a critical advance because that enabled us to build this machine that can now do extremely high-throughput single-cell genome sequencing. It led to the creation of my company, GnuBIO. The two postdocs who got this to work, Adam Shiambi and Dennis Eastburn, they solved that problem, and they were able to create a company. They're now the co-founders with me of this company to actually commercialize that technology. So I guess it's a challenge, it's a struggle, but it's also potentially an opportunity. But it is something that we've often find, which is these problems that are sort of finished, but not really. And they need to be totally finished in order for us to move forward. And it's a bit of a thankless job. But when you're building technologies, often this is unavoidable because you just can't progress until you solve these issues.
Definitely. And I think as technology advances, you discover more and more that some of these questions that we thought were answered are not answered as well as they could be, because maybe you didn't even have the technology at the time that could answer it to the level you're doing now.
Oh, absolutely.
Well, phenomenal to hear about one of these challenges that you're facing right now in your career. And I think, like I said, it's so important to share these with people, but I think it's also important to take time to celebrate your successes. So Adam, I want you to tell us next about one of your own success stories, whether it was a small win or just a huge victory that meant a lot.
Well, I mean, again, I think sort of like failure, victory, relatively, it's much rarer, but it still happens on a regular basis. I mean, there's the many small victories when the student's experiment works finally after the 9th time, on the 10th time it works. Mm-hmm. I would say that the single cell sequencing, it's a fairly recent success that I'm very, very pleased with and just so happy, because it was the result of this multi-year long struggle on this mundane problem that really wasn't recognized, and probably very few people care that we solved it, that then led to the development of this really groundbreaking technology that now has allowed us to create a whole company to commercialize it. That's one of the biggest scientific successes that I've had very recently that I'm proud of.
Well, congratulations on this success. And can you tell us, do you remember the moment where it looked like this single cell sequencing was fixed?
That's funny. The reality is that you never have that moment. What it is that science is, or maybe the natural world sort of is inherently deceitful. Mm-hmm. Maybe it's the human mind, is that you want to be successful. And so when you see successes or things that look like success, I think people sometimes jump to conclusions too quickly they were successful. And what ends up happening is they weren't, and then you sort of have a crash. So I'm always nervous when we're successful. And it never stops. I just get less nervous as more data comes out and supports the previous data. So I wouldn't say that it ever is a moment where, that started working.
That's it. Yeah.
I mean, it did happen where we solved the problem. And there was that moment. I didn't recognize it at the time. At the time, I was like, okay, this looks good, but
let's try again.
Let's do it 10 more times and see. And after, several more months, there's like, okay, well, everything is holding together. And you just build more and more confidence. And that really is how science works ultimately, right? I mean, a scientific theory can be disproven at any point, even no matter how many hundreds of years of evidence has supported that it works, right? So think of Newtonian physics. That stood for hundreds of years, literally. And overnight, it was proven wrong by Einstein, and then more by the development of quantum mechanics. And so that's a common challenge that even when we're successful in the lab, you only ever really recognize your success sort of far hindsight when it's lived long enough that you're pretty confident that it's true.
Absolutely. Well, I think that makes it almost exciting to be in science, because you never know what direction things are going to go. You never know what thing that's been well established and accepted is going to, like you said, overnight be found to be not true. So phenomenal to hear about one of these successes. And I know we've talked a little bit about your life outside of science and how you spend your time, but I love talking about what scientists are reading. So do you have a book recommendation for me and our listeners that you can share with us, whether it's a science book or a non-science book?
So I actually don't read very much in terms of books. I read so much science papers that I actually, in my free time, I try to listen to things, usually while walking. So a lot of podcasts, and one in particular is Dan Carlin's Hardcore History. I don't know if you've heard of that, but
yeah,
it's an amazing series done by just an individual, Dan Carlin. He's just really a gifted historian, the way he talks about history. I've listened to all of his histories probably 3 times now. I love him so much. Another is there's a course series by a physicist at Stanford named Leonard Susskind called The Theoretical Minimum. And it's something actually I watch pretty regularly because I don't get to do physics anymore as a professor, basically doing bioengineering. So for those of you who have sort of an intrigue for physics and wish you understood it more deeply than the level of just popular science, these lectures by Leonard Susskind are really amazing because I got a PhD in physics and he really pulls no punches. I mean, he simplifies this a little bit and he condenses the material down, but he really tells you how it really is and how physics is really done. And he has a whole series on all the different branches of theoretical physics that's amazing.
I also love a podcast called This Week in Virology. It's been really educational for me. Vincent Racaniello's podcast. His other ones, TWIV, TWIP, and TWIM. TWIV is my favorite, although I usually fast forward past the weather part.
Right.
But actually, one of my close collaborators, Ken Stedman, the collaboration began because I heard him on This Week in Virology, and he was talking about his science, and I immediately had an idea that we could help him. And I emailed him, and that created a 3-year collaboration that actually just resulted in 3 new papers that have just been published.
Oh, that's awesome.
And then the last thing I would say is that there's a really excellent YouTube video by a professor at University of Central Florida named Kenneth Stanley called Why Greatness Cannot Be Planned, The Myth of Objective. And I think more than anything else that his philosophy that he describes in this YouTube video really captures what I think how science really works. This idea that it really is us stumbling around in the dark into incredible discoveries. And trying to specify an objective a priori and then hit that objective is almost guaranteed to lead to failure in science. So if your goal is to extract the most value out of science, you just have to walk around in the dark a lot and be open to any opportunity that arises. And I think that's really how science works and why basic science research is so vitally important to the continued advance of our species and the continued economic potential of our country. So he actually has several videos on YouTube where he describes this idea, and I think it, more than anything else, really describes how I believe science to work.
Well, Adam, excellent recommendations. I will put them out on our website for our listeners to find if they're looking to be entertained and learn more about science. And as a scientist, you get a lot of opportunities not only to work on amazing things and work with amazing people, you also get to travel. And I think there are so many opportunities for this as a scientist between, like you mentioned, working with collaborators, also presenting your work. Is there one place that really sticks out in your mind, Adam, as somewhere that science has taken you that was just your absolute favorite place?
The first time I went to Asia, Japan, China was for science, and that was incredible. Of course, all the different trips to Europe have been fun. Most recently, maybe it sticks out because it was very recent, was I went to Hawaii for a conference where many of my colleagues were there, people that I've known now for over a decade. And it was just great to be in this beautiful place in the big island of Hawaii. Everyone wearing Hawaiian shirts and shorts and flip-flops and listening to science lectures. Plus, my wife came with me, so we got to go scuba diving for the first time. And,
awesome.
And we saw lava. Apparently, that lava area slid into the ocean, so I guess it was good we weren't there.
Oh, geez.
Most recently. But yeah, that was a very, very cool trip for all of these different new experiences that we had.
So what did you think of scuba diving? Would you do it again?
At first, I thought I'd be terrified. Mm-hmm. I grew up in Miami, so I should like the beach. But in reality, the ocean, as I've always found it a little bit scary, just this massive, dark, cold water full of monsters. But I actually really enjoyed it. Once I got under the water, it was really calming. And I think much of it is that in order to control your buoyancy, you need to control your breathing. So it forces you to breathe in a very controlled manner, take very slow, careful, controlled breaths. And that naturally makes you calm. And so it's not something I normally do. I think I'm typically a pretty high energy person. So being 50 feet underwater, breathing extremely calmly and regularly and seeing the most incredible sea animals. There were no monsters that day.
Right. You got lucky.
But just fish, and literally a sea turtle on my first dive swam 1 foot from my face and looked at me. So it was pretty amazing. It was pretty amazing. I didn't think I'd like it, but I really did. And it turned out that the idea of breathing underwater with 50 feet of water above your head, it was a little scary, but it was pretty easy to not be constantly worried about when you're down there. You sort of just get totally into what the amazing things that are around you.
Absolutely. Well, Adam, this sounds like a phenomenal experience. Thank you so much for sharing it with us today. And I think we've talked about some of the people that you've encountered over the course of your career as a scientist. And I think it's fun to talk about these people because there's so many different personalities in science. And I think there are some stereotypes out there about what scientists are, quote unquote, like, what they look like, what they act like, what they do. And I think many of these are wrong. So I love talking about the quirky traditions and the fun things that scientists do when they're together. So do you have a story of your own that really showcases this human side of science?
Actually, my lab, I would say we're just regular people, not particularly quirky. I think for the most part, maybe many people think that science is sort of run as a dictatorship and people run their labs where you have this genius professor who's telling everybody what to do. And really, that's not the way it is, at least not in my lab. I try to make it absolutely flat. The amount of seniority or perceived seniority that any person gets is really going to be earned by them being exceptionally good and just earning the respect of their peers. I really ask everybody to question everything we do because I'm always so fearful of us being deceived by a false result. And that includes myself. So I try not to run things at all like a dictatorship and try to keep it as flat as possible and have very open and clear discussions with people. So I don't know if that's how non-scientists see science, but I think that was a little bit of an eye-opener for me. Actually, many labs I've been in are that way. Everybody can challenge anybody, including the professor at any point. There's not this very rigid hierarchy that people might think exists.
Yeah, it's a little scary, though, the first time you find yourself questioning your advisor. You're like, am I going to get yelled at? No, it's fine.
Yeah, no, I don't think my people have any fear of questioning me.
Oh, good.
Honestly, I love it when they question me. I always have a feeling of pride when, usually it's true that especially the very young students, the first year, second years, they're gun shy, but it's when that student for the first time questions something I said was true. I have a moment of pride where I'm like, okay, this is what it means to be a mentor. This person is developing and recognizing that I'm just another person. Mm-hmm. I'm no smarter than this student is. And they can do what I can do, and they can question my ideas and have their own great ideas. And it's amazing to see that transformation now. I've seen it a few times now because I've had several students. And there's this point, not all of the students go through this, but many of them do go through this, where there's this point where they start to think like an adult's not the right word, but like an adult scientist, like somebody where they start to believe their own intuition and their own judgment, even over somebody who's telling them something that is true when maybe they should defer to them, but they don't. I always have a moment of pride when my students do that.
Definitely. Well, it sounds like you've got a great collaborative, collegial environment too, where you are encouraging people to be confident and assertive scientists. And I think that's so important.
Oh, absolutely. I mean, the worst thing in science is to be deceived by your own belief that something is true that isn't. And we are all susceptible to that. And the best safeguard against that is to listen to everybody. Don't rely on any one person.
Absolutely. And we've talked a little bit about some of the work that you're doing in the laboratory and some of the amazing technology you've been able to develop. But I'm curious, Adam, if we gave you everything, the sky's the limit here, funding, staff, technology, feasibility, whatever you want, what is the one thing you would want to develop or the one question you would want to answer?
It goes back to the vision of my lab, which is this idea of being able to completely store all of the information that encodes a living system. So I'll start off with just cells. Being able to have the whole genome of a cell, all of the RNA molecule sequences, all of the proteins that are present and their different modifications, a set of all of the molecules that are present. All of this information about what the cell is composed of, being able just to get that information is going to take an enormous investment in technologies that I think is critically important to the advance of science. That's something I would love, that I want to spend my career working to accomplish. And then I'm hoping that it'll be done in a time, I'm still relatively young, so I'm hoping it'll be done in time where I can also be part in thinking of how to turn that into a computer model that can actually faithfully represent a living cell. That's my overarching goal.
I love it. So if you could start with any cell, what kind of cell would you pick?
I'm sort of cell agnostic, I guess, because ultimately we can't even understand one, let alone all of them. But visceral example of where this would be valuable is in cancer. Cancer is a disease of the genetics of cells. Their genetics mutate, their genomes mutate, and you end up having this sort of hyper-evolution in a tumor where all the cells are competing with one another for survival. They're literally competing with one another for survival, for resources. And that is what leads eventually to cancer becoming deadly. And we just do not understand how that process works in general. We understand very broadly that it's this process of evolution, but how does it actually work? How can we treat a tumor, a particular person's cancer? These are questions that are going to really require us to go into that tumor and trace the evolution of that particular person's tumor and figure out how did these different populations of cells arise? What's their potential for eventually spreading through the body, or if I give a treatment, evolving to resist that treatment? We're never going to be able to do that really effectively until we can faithfully write down all of the aspects of the tumor that are relevant. So this comes back to being able, it just starts with being able to characterize what are the cells of that tumor composed of, and then repeating that on the millions of cells that are inside of even a very small tumor.
Gotcha. Well, Adam, this sounds like a phenomenal goal to be working towards over the course of your career. And I know we've talked about some of the different stages that you've gone through in your career, some of the different people you've met along the way. Is there any one piece of advice that you received at one point that really helped you that you want to pass on to our listeners today?
Yeah, I think I would go back to the Steve Jobs quote, which is that all the incredible technologies and things you see around you were built by people just like you. They weren't any smarter. They didn't have a magical ability that you don't have. They were just people who put in the effort to understand whatever they were trying to create. They put in that work and they did it. And it's an ability that we all have. And I think once you recognize that, it makes you much more willing to take on challenges that seem insurmountable. But then when you get into it, like figuring out how to install lighting in your kitchen, you realize it's really not that hard. And it sounds like a trivial example, but honestly, most things in science that we've discovered and developed were not much harder than, for me, figuring out how to go into my attic and connect to our old knob and tube wiring in our house. Mm-hmm. So that's a piece of advice I would give.
Well, I love this analogy that you've drawn from science to renovating your home. And I think you're absolutely right here. I think on the surface, science and scientists can seem really intimidating, but especially for all the young scientists out there, you too can be in the future making these important discoveries. And it's regular people, like you said, who are doing these things. So phenomenal message to leave our listeners with. And is there any other last piece of advice you want to give them, or a last note of inspiration you'd like to end our conversation on today?
For me personally, what is actually inspirational is to be just very grateful for the opportunity that I have as a scientist to follow my own ideas and to have it paid for by taxpayers. And I think similarly, it's also inspirational. It's probably more motivational, which is that I feel, and I think as scientists, we all have an enormous responsibility to deliver on an investment. Science is not cheap. It's an extremely expensive endeavor. There's so much failure. And with it becoming more reliant on technologies, it's becoming more expensive. And we shouldn't take that lightly, the amount of money that the taxpayer is investing to allow us to have this amazing opportunity. That would be my advice, would be to be grateful and feel some responsibility to actually the people that are making it possible for you to do what you want.
Well, wonderful message to share. And again, thank you everyone out there who is supporting science in whatever way you can. And Adam, can you tell our listeners now how they can reach out if they want to learn more about you and what you do?
Oh, yeah, sure. I mean, we have a lab website. It's www.abatelab.org. And of course, you can always just email me. My email is adam@abatelab.org.
Perfect. Well, listeners, definitely get in touch if you have questions for Adam. And Adam, thank you so much for joining us on the program today.
It's my pleasure. It's been a lot of fun.
Well, it was great to speak with you. And listeners, wonderful to have you here with us as well. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.